Hello everyone. Today we are going to discuss about the testis, epididymis and scrotum, which are the parts of male reproductive system. So here is the sagittal section of male pelvis where we can appreciate the internal organs, that is reproductive organs. So we can see the testis in the scrotal sac, and testis continues to form the epididymis, which is on the posterolateral aspect of the testis, and epididymis continues from its lower pole as a vas deferens. So vas deferens, this muscular tube, we can see it passes through the seminal vesicles. Seminal vesicles is a gland, and here we can see finally the vas deferens passes through the gland called as prostate. So it is the prostatic part of urethra is here, which is surrounded by the prostate gland, and vas deferens finally terminates into the prostatic part of urethra. So this is a basic idea about the internal male reproductive organs. So testis, epididymis, vas deferens, seminal vesicles and prostate. Testis develops retroperitoneally in the abdomen and descends into the pelvis and eventually reaches the scrotum. So we can see here the testis is present in the scrotum, and due developmentally, if we see, it develops in the abdomen on retroperitoneally, means behind the peritoneum over the posterior abdominal wall, and later it reaches the scrotum, and scrotum contains the testis and epididymis, and here it maintains at a temperature 2 degrees lesser than a normal human body temperature, that it is maintains around 35 degrees. So scrotum contains the testis and epididymis, which regulates the temperature of the testis and maintains at 35 degrees Celsius, which is in Fahrenheit 95 degrees Fahrenheit, and that is around 2 degrees lesser below the body temperature of 37 degrees Celsius or 98.6 degrees Fahrenheit, which is essential for the requirement of spermatogenesis. So lower temperature is very much essential to perform spermatogenesis. So continuing with the scrotum here, the scrotum, we can see the layers in this image where the skin, just beneath the skin, there is a muscle which is called as dartos muscle. So this one is the dartos muscle, which is present subcutaneously in the skin, which wrinkles the skin, and it continues with the superficial penile fascia and superficial perineal fascia. So dartos muscle, actually it is the continuation of superficial fascia of abdomen, and it continues in the perineum as superficial perineal fascia and around the penis as superficial penile fascia. And next to the dartos muscle, the next layer is the cremaster muscle. So here is the cremaster muscle. So the cremaster muscle, it is the continuation of spermatic fascia, and where the cremaster muscle covers the scrotum, and upon the contraction of cremaster muscle, it elevates the testis. So that is called cremastric reflex. And if we talk about the arterial supply of the testis is mainly by the external pudendal arteries and posterior scrotal arteries, which are the branches of internal pudendal arteries. Posterior scrotal arteries are the branches of internal pudendal arteries, and also external pudendal arteries supplies the scrotum. Nerve supply: anterior scrotal branch of ilioinguinal nerve, genital branch of genitofemoral nerve, posterior scrotal branch of that is the branch of pudendal nerve, and perineal branch of posterior femoral cutaneous nerve. So these are the nerves which supply the scrotum. So let's see the comparison of layers of the scrotum. So the skin is the continuation of the skin of anterior abdominal wall. Superficial fascia of the anterior abdominal wall continues as dartos muscle, and we saw the dartos muscle continues in the perineum as perineal fascia, a superficial perineal fascia, and around the penis as superficial penile fascia. And external oblique muscle continues as external spermatic fascia. Internal oblique muscle continues as cremasteric muscle and its fascia. And transversus abdominis doesn't have any corresponding layer. It is not having any corresponding layer. So one thing we have to note here, the transversus abdominis muscle does not continue into the scrotum. And whereas the fascia transversalis is the fascia in the abdomen beneath the transversus abdominis muscle. This fascia transversalis continues as internal spermatic fascia. It continues as internal spermatic fascia around the testis. So these are the coverings, that is the scrotum, layers of the scrotum, and how the abdominal wall layers continues into the scrotal wall layers. So let us discuss about the testis and its coverings now. And it is surrounded incompletely, especially on the medially, laterally and anterior aspect, but not on the posterior aspect. So this covering is like a sac, peritoneal sac, which is called as tunica vaginalis. So we can see this peritoneal sac, which is getting reflected all around, is the tunica vaginalis, and it is a double layered, just like the other peritoneal reflections. So the inner layer is the visceral layer of tunica vaginalis, and the outer layer is the parietal layer of tunica vaginalis. So tunica vaginalis is the pouch of serous membrane that covers the testis, and it is derived from the processus vaginalis of peritoneum, that is in the abdomen. When the testis descends down, it brings a part of peritoneal sac around with it, which is called as processus vaginalis, which later turns to form tunica vaginalis in adults. So within the fetus, the precedes the descent of the testis from the abdomen into the scrotum, and it is the serous sac of peritoneum that covers the front of the testis and epididymis. It consists of parietal layer that forms the innermost layer of the scrotum, and a visceral layer which is adherent to the testis and epididymis. So we can see the visceral layer which is along the testis and epididymis as well. We can see it is reflecting around the head of the epididymis. And here it's the cut section of seminiferous tubule is shown, where we can see the various cells present inside the seminiferous tubules. So the processus vaginalis is an embryonic diverticulum of peritoneum, and that traverses the inguinal canal, accompanying the testis in its descent into the scrotum. So like it is a round ligament in females which is present in the inguinal canal, and but here instead of round ligament, the spermatic cord along with the processus vaginalis, that is a part of peritoneal sac, it settles into the scrotum, and later it gets obliterated and separates from the rest of the peritoneum, and it closes completely, forming a closed sac called as tunica vaginalis in male. And persistence of processus vaginalis leads to the development of congenital indirect inguinal hernia. But if its middle portion persists, it develops into congenital hydrocele. So in previous slide we saw the tunica vaginalis. Beneath the tunica vaginalis, the thick connective tissue which is like a capsule around the testis is tunica albuginea. So tunica albuginea, it surrounds at the testis like a thick capsule, and beneath the tunica albuginea there is a highly vascular layer which is called as tunica vasculosa. So beneath the albuginea, the deeper layer is the tunica vasculosa, which is highly vascular layer of connective tissue. Then about the tunica albuginea, here it sends septa. You can see here it sends septa into the substance of the testis. So tunica albuginea, when it sends septa towards the mediastinum, this is called as the mediastinum of the testis, where we can see the ducts are forming. That is called as this plexus like thing is called reti testis, and this area is called as mediastinum testis. So we can see the septa towards the mediastinum testis, dividing the testis inner aspect into the several compartments. So the tunica albuginea, as it projects inwards towards the mediastinum, divides the testis in about 250 lobules, 250 small compartments, and each of which contains 4 coiled seminiferous tubules, 1 to 4. Some compartments may have a single seminiferous tubule, some may have up to 4. So 1 to 4 highly coiled seminiferous tubules are present in each compartment, and these septa converge towards the midline of the posterior surface, and where they meet to form a ridge-like thickening known as mediastinum. So we can see on the posterior side, this is the posterior aspect where I have shown the mediastinum, where it contains the reti testis. So that was about the layers surrounding the testis. So testis mainly it contains tubules. So here is an histological image to show the tubules of the testis, which are called as seminiferous tubules. And seminiferous tubules contain, in between these tubules we can see the clusters of cells which are called as Leydig cells or interstitial cells. So interstitial cells of Leydig are present in between the seminiferous tubules. Inside the seminiferous tubules of the testis, we see the several cells present within the seminiferous tubules, and in the lumen we can see it is quite thread-like appearance, which are formed by the tails of the sperms. So these cells which are present inside the seminiferous tubules are spermatogonial cells, and which are adjacent on the inner side of the tubule, and they divide in a centripetal direction and beginning in the walls and proceeding towards the lumen of the seminiferous tubule to produce the sperm. So sperms will settle in the lumen, and we can see they are still intact with the spermatogonial cells, and in between the spermatogonial cells there are supporting cells, a large supporting cells which are called as sustentacular cells or Sertoli cells. So that is a little bit about histology. So seminiferous tubules and Leydig cells are present. Next, regarding spermatogenesis, the whole process of production of the sperm is called as spermatogenesis, and it takes about 74 days to complete, and it is around 200 to 300 million spermatozoa are produced daily, but about half of them becomes a viable sperms, where the Leydig cells secrete hormones which are called as testosterone hormones, and not only testosterone, androsterone and dihydroepiandrosterone, that is called as DHEA. So all these are produced. So we can see here, usually it takes around 74 days, around 200 to 300 million sperms are produced, and half of them only becomes liable, and testosterone is produced by the Leydig cells. So we can see this is the wall of the seminiferous tubules, and this large cell, large columnar or pyramidal shaped cell we can see here. This is the Sertoli cell. We can see its nucleus here. This is the large nucleus present on each side, and in between the Sertoli cells we see the developing of these small cells which are called as spermatogonial cells, and initial division of spermatogonial cells is by mitotic division, and later they divide it to enter into first meiotic division. First meiotic division, we see the chromosomal number becomes haploid, and then further the cells enter into second meiotic division, where the haploid number chromosomes are continues as haploid. It is similar to mitosis, second meiotic division, and we can see the formation of spermatids here at the end of second meiotic division. Spermatids are the round cells, and these spermatids further divide to form differentiate to form a sperm having a tail, body, and neck and headpiece. So it forms a complete sperm. So the formation of spermatid to sperm is called as spermiogenesis. So that is about the in short about the spermatogenesis. So if we see the sequence of movement of the sperm, we saw the sperm is produced in the seminiferous tubule. So here it in the seminiferous tubule, within the lumen we have the sperms, and these sperms will enter into the straight tubules, and we can see here these are the straight tubules, and straight tubules continues to form a meshwork like structure called as retitestis, and retitestis continues joins to form like efferent ductules. Usually some 10 to 12 efferent ductules are present, and we can see the efferent ductules, they are opening into the epididymis. They are opening into the epididymis, and so that is the way where the sperm will transfer from the seminiferous tubules to the retitestis, and from the retitestis to the efferent ductules, and from the efferent ductules to the epididymis, and from the epididymis to the vas deferens.